Welcome to this section on the development of the brain. So, brain development depends upon maturation and learning. So, the learning that happens in early stages of development, and maturation meaning the brain's wiring of itself and how that changes during early development. I'm going to talk about how neurons develop, how their axons connect, and how experience modifies development. So, the human central nervous system begins to form when the embryo is approximately 2 weeks old. So, we saw a figure describing the the change from the neural tube into the three major divisions of the brain. So the dorsal surface thickens, forming a neural tube. The tube surrounds a fluid filled cavity. The fluid's the cerebral spinal fluid, and this cavity becomes the spinal um, column. The forward end enlarges and differentiates into the hindbrain, midbrain, and forebrain, the hindbrain developing earlier. And as the embryo is developing, we get the midbrain and then the forebrain sitting on top of that. And again, the rest of that neural tube becomes the spinal cord. So here is one figure. Here we have the brain and spinal cord begin as folding lips surrounding the fluid-filled canal. This stage is approximately 2 to 3 weeks after conception. And now we can look at the development of the brain over these 4 stages. So here at 3 weeks, we are starting to see the differentiation of the hindbrain, the midbrain, and the forebrain. And we can see the enlargement of these areas. And we can start to see the forebrain surrounding parts of the midbrain and hindbrain. Right here we have the brain stem. Oops. And then by birth, we see the forebrain with its gyri and sulci developed in some areas, not the prefrontal cortex, but areas that are receptive to sensory information are beginning to be, beginning to be developed at birth. So the fluid-filled cavity becomes the central canal of the spinal cord and the 4 ventricles of the brain. So remember that the cerebral spinal fluid moves through the ventricles and the spinal cord. It provides a reservoir of nutrients for the brain. It provides cushioning for the brain during head movements. Now, at birth, the human brain weighs approximately 350 grams. But by the first year, the brain weighs approximately that of the adult brain. So intelligence is not just a function of brain size. As you can see, by the first year, the size of the brain, the weight of the brain is that of the adult. However, we are continuing to learn. Our frontal cortex especially is continuing to develop. So this is going to be a function of the connections that are made among neurons and the organization of particular areas of the brain. And I love that your textbook author cues you into thinking about the learning that happens over the first year of life that is also correlated along with the development of the brain. But there's also learning that happens all throughout our development as human beings that are going to affect the organization of our um, neuronal systems of communication, neuronal networks, I should call them. Okay, so there are 5 stages in the development of neurons. We have proliferation, migration, differentiation, myelination, and synaptogenesis, which is my favorite. I just love the word. So let's talk about each one of these. So proliferation is the production of new cells, neurons in the brain primarily occurring early in life. So early on in development, there are cells that are lining the ventricles and they divide. And some cells become stem cells that continue to divide. And others become neurons and glial that are going to then migrate to other locations. So migration is the movement of the newly formed neurons and glial to their eventual locations. Some do not reach their destinations until adulthood. Other neurons, like those in portions of sensory systems, migrate quite early on in development. Motor neurons develop early on as well. So migration occurs in a variety of directions throughout the brain, and chemicals work to direct the migration of neurons. Chemicals such as immunoglobulins and chemokines are going to guide the movement. Differentiation is the forming of the axon and dendrites that give the neuron its distinctive shape. So remember that neurons in different areas of the brain have different shapes. They have different structures to their receiving parts or their dendrites. And we have neurons that have axons of varying lengths depending on where in the body they're found. Now the axon grows first either during migration or once it has reached its target, and is followed by the development of dendrites. So the question becomes, how does an axon reach its target? I'm going to talk about that in a moment. It's going to be chemical gradients and chemical traces that attract those axons to their targets. Myelination is the process by which glial cells produce the fatty sheath that covers the axons of some neurons. So remember that the glial cells in the central nervous system that create the myelin are oligodendrocytes. The glial and the peripheral nervous system that creates myelin are called Schwann cells. And the function of the myelin is to speed up the transmission of neural impulses. And remember that at points in the axon, at points along the axon in the myelin, there are gaps. And those are called the nodes of Ranvier. And there are sodium channels at those gaps, so the action potential can regenerate as it moves down the axon. Now, myelination first occurs in the spinal cord and then in the hindbrain, midbrain, and forebrain. And this occurs gradually for decades. And synaptogenesis is the final stage of neural development, and it's the formation of the synapses between neurons. And this occurs throughout life as neurons are continually forming new connections and discarding old ones so that use it or lose it. You've probably heard that phrase multiple times. Neurons are consistently forming new neural networks Think about habits or automatic behaviors. Those develop because there is communication between particular neurons. They form networks and these synaptic connections. The process of synaptogenesis does slow significantly later in the lifetime. Now, when we are first developing, the nervous system makes many more neurons and connections than are what are needed and what are going to be kept. Now let's talk about new neurons later in life. So it was originally believed that no new neurons were formed after early development, but later research suggests otherwise. And we've talked about this already. There are two areas within the human nervous system where we see new neuronal growth or neuronal generation. And one of those areas is in the olfactory system. The olfactory receptors are cells that are exposed to the external environment. So they're exposed to toxins in the environment and they can be damaged. They are going to regenerate themselves. So new olfactory receptors continually replace dying ones. The other area of the brain where neurons regenerate are the hippocampus. Stem cells are going to differentiate into new neurons in the adult hippocampus of mammals that facilitate learning. We see in songbirds that there's a steady replacement of new neurons in the singing area of the brain every spring when the songbirds begin to sing. Now, stem cells, by the way, are undifferentiated cells that are found in the interior of the brain, and they divide and they produce daughter cells that can transform into glial cells or new neurons. And again, in the human central nervous system, the olfactory receptors can regenerate, and so can cells in the hippocampus. Okay, so different cells have different average lifespans. Skin cells regenerate most frequently. Most are under a year old. Heart cells tend to be as old as the person is. And the mammalian cerebral cortex form few or no new neurons after birth. Again, the exception is olfactory receptors and cells in the hippocampus to facilitate learning, memory formation. Okay, so we were talking about migration, and I said that how is it that axons are going to find their target? Because they need to travel a great distance across the brain to form the correct connections. And so Sperry's research with newts indicate that axons follow a chemical trail to reach their appropriate target. Growing axons reach their target area by following a gradient of chemicals in which they are attracted by some chemicals and repelled by others. So what Sperry did is he severed a portion of the optic nerve. He severed the part of the optic nerve that makes contact with the thalamus. So remember, the thalamus is the sensory relay station. It is the site of reception of sensory information before being sent to the cortex. So information for vision is going to go from the retina through the optic nerve to the thalamus. Sperry cut that connection. Now in the newt, these cells, these neurons can regenerate. And what he found was that these cells regenerated and they grew back to their original target. So in the next set of experiments, what he did was he severed the connection of the optic nerve to the thalamus, and he rotates the eye 180 degrees, and he sees that the dorsal portion of the retina, the axons grow back to their original target, the dorsal portion of the thalamus, ventral to ventral, again by following the chemical makeup, the proportion of particular chemicals in the axon will match that proportion of those chemicals in the thalamus. So unfortunately, this animal is now seeing the world upside down, but we get to learn something about axons and how they reach their targets. Right, so they grew back to their original target, not the target corresponding to the eye's current position. Again, they do that by following a chemical gradient. So again, there's different concentrations of proteins in the dorsal retina and the ventral tectum. So axons that are rich in the protein attach to the neurons that are also rich in that chemical. And that's how they found their way back to their appropriate or original targets. And again, the result is that this animal is now encountering the world upside down. So the general rule is that there is competition among axons. When axons reach their targets, they form synapses with several cells. The postsynaptic cells strengthen connections with some cells and eliminate connections with others. The formation or elimination of these connections depends on the pattern of input from incoming axons. So again, this maps onto the use it or lose it principle of synaptic connections or neuronal networks. There are some pathways that are going to be used more frequently than others. That synapse is going to remain, and other ones are going to degrade and cells are going to die. And some theorists call this neural Darwinism, this selection process of neural connections. And again, it's this competition among synaptic connections. So early in development, we initially form more connections than we need. And the most successful connections and combinations are going to survive, while the others fail to sustain active synapses. So Levi Montalcini discovered that muscles do not determine how many axons form, they determine how many survive. So again, during development, we produce more neurons and synaptic connections than what is needed. And so the brain overproduces neurons that have a built-in suicide program called apoptosis. So when neurons communicate in early development, nerve growth factor is released. And this chemical, this protein, is going to promote the survival and growth of axons. So if a cell is not releasing, if a cell is not receiving nerve growth factor, the cell is going to move into its program of cell death, which again is apoptosis. Right, so cells need to be exposed to neurotrophins after making connections, or they undergo apoptosis, which is a pre-programmed mechanism of cell death. So healthy adult nervous system contains no neurons that fail to make appropriate connections. So again, that myth of we only use 10% of our brains is just that, a myth. Right? All of the cells that are still alive are cells that have functioning networks. Nerve growth factor is a neurotrophin. So the elimination of massive cell death is part of normal development and maturation. After maturity, the apoptic mechanism becomes dormant. The visual system is actually thicker in blind people due to a lack of visual stimuli, so it cannot prune out ineffective neurons. Now, if you follow this link, you will, it's another flash player. Okay, so that link is actually not going to work. But what was here, if you get access to Adobe Flash Player, it shows you the difference in appearance of apoptosis and necrosis. So apoptosis is the programmed cell death, how the and then the microglial coming clean up the cells. They're neatly packaged so that the microglial can clean up the debris. Whereas in necrosis, which is cell death due to assault or toxins, it's not a neat packaged cell death. There is debris that is more spread out that the microglial then have to deal with. Okay, so the developing brain is vulnerable, and early stages of brain development are critical for normal development in later life. We talk about critical periods. The early stages of brain development are critical. So if there's a mutation on one gene, it can lead to many defects. Chemical distortions in the brain during early development can cause significant impairment and developmental problems. Fetal alcohol syndrome, for example, is a condition that children are born with if the mother drinks heavily during pregnancy. And the influence of drinking at different stages of pregnancy are outlined in the text. And you can see how the effects are much worse when it's earlier on in development. But the behavior of the mother is going to influence the behavior of the offspring. And this is going to be the case whether or not it's alcohol ingestion, or if it's stress or something else. So the effects of the mother drinking heavily during pregnancy is hyperactivity and impulsiveness, difficulty maintaining attention, varying degrees of mental retardation, motor problems and heart defects, and facial abnormalities. The dendrites of children born with fetal alcohol syndrome are short with few branches. Exposure to alcohol in the fetus brain suppresses glutamate and enhances the release of GABA. So suppressing glutamate, that's an excitatory neurotransmitter, while enhancing GABA, the inhibitory neurotransmitter, means that there's less activation of neurons in the central nervous system. And again, if neurons do not release nerve growth factor, they are not going to maintain their synaptic connections. So many neurons receive less excitation and exposure to neurotrophins than usual and undergo apoptosis. Okay, so again, neurons in different parts of the brain differ from one another in their shape and chemical components, and therefore their functioning. Immature neurons transplanted to a developing part of the cortex develop the properties of that new location. Neurons transplanted at a later stage of development develop some new properties but retain some of the old properties. So we can see this in the ferret study. So first, the normal right hemisphere is trained to respond to a red light by turning to the right. Then the rewired hemisphere is tested with a red light. The fact that the ferret turns to the right indicates that it regards the stimulus as light, not sound. Fine-tuning by experience. So the brain has some ability to reorganize itself in response to experiencing, or in response to experience. So this is, this is related to the nature-nurture debate. Is it all genes or is it learning? And the brain does reorganize itself in response to experience and response to learning. Axons and dendrites can modify their structure and connections throughout the lifetime. Dendrites continually grow new spines and make new synaptic connections. The gain and loss of spines indicates new connections, which relates to learning. Okay, so here is a figure that is detailing the changes in dendritic trees of two mouse neurons. So during a month, some branches elongated and others retracted. So here we can see elongated spines. Here we can see retracted spines. So, an enriched environment will stimulate more neuronal branching than a non-stimulating or an impoverished environment. So as you can see, these are the dendritic spines in a jellyfish that was reared in isolation. Develops neurons with very few branches, whereas this is a dendritic tree of a jellyfish raised in an enriching environment. Now, rats raised in an enriched environment develop a thicker cortex, increased dendritic branching, and improved learning. Measurable expansion of neurons has also been shown in humans as a function of physical activity. As old neurons die by apoptosis and new ones form to take their place, there is improved learning and memory. I didn't mention this earlier, but if we talk about the behavior of rat mothers and the behavior of their offspring, if we have a rat mother that has been exposed to a lot of stress, they are going to attend to and groom their offspring much less, which is going to result in offspring that has more anxiety or fearful responses to multiple stimuli. And something very similar happens in human beings as well. Right? So mothers who experience stress and an inability to emotionally regulate themselves have a very difficult time attuning with their young, providing the the appropriate or correct cues to the to the to the needs, the safety needs of their young, which can result in a hypervigilant nervous system. So it was once believed that teaching a child a difficult concept would enhance intelligence in other areas. This concept is known as far transfer. Evidence shows that skills associated with practice tasks transfer, but not other skills. The brain cannot be exercised like a muscle. Effects of special experiences. So blind people improve their attention to touch and sound based on practice. Touch information activated the occipital cortex area, which is ordinarily devoted to vision alone. So if you do experiments with blind people and you have them read Braille, areas of the occipital lobe will show activation in an fMRI scan. So the occipital lobe, normally dedicated to processing visual information, will adapt to process tactile and verbal information as well. Right? So you have this whole area of the brain is not going to shrivel up and based on there's no visual experience. It's going to adapt. It's going to be enrolled to process other types of information. People blind from birth are better at discriminating between objects by touch and have increased activation in their occipital cortex while performing touch tasks like reading Braille. Further research using magnetic stimulation to inactivate brain areas demonstrated that blind people use the occipital cortex to discriminate between tactile stimuli and Braille symbols, but sighted people do not. So when you read the module on research methods, you're going to read about transcranial magnetic stimulation. So there is a machine that has coils that you can send pulses of magnetic stimulation to the brain. When you do that to the occipital lobe while blind individuals are trying to read Braille, you disrupt that functioning. And then we conclude there's a correlation between touch information in blind individuals and the occipital cortex. You do that same experiment with sighted people and you don't get the same results. So we can talk a little bit about music training. So MRI studies reveal the following. The temporal lobe of professional musicians in the right hemisphere is 30% larger than non-musicians. thicker gray matter in the part of the brain responsible for hand control and vision of professional keyboard players. And results suggest that practicing a skill reorganizes the brain to maximize performance of that skill. So the more you engage in a particular behavior, the stronger that network, the stronger that connection becomes, right, among cells in the network. Okay, so here is just a figure that shows the areas of the cortex involved in musical practice, where their areas in red have the thicker gray matter among professional keyboard players than in amateurs, and thicker among amateurs than in non-musicians. Areas marked in yellow showed even stronger differences in the same direction. Adult experiences can also modify brain anatomy. However, research is needed to determine whether the effects are strong enough to be observed with MRI or similar technology. So I can tell you that recent research is showing that meditation is thinning the amygdala. It's that area that plays a role in discerning emotional information, especially that concerned with fear, thins. So our fear responses become lessened. We can make finer discriminations in the environment in which we respond with fear. So meditation will thin the amygdala, but it will thicken the prefrontal cortex, the areas involved with planning, abstract thinking, decision making, impulse control. Those areas and others are affected by meditation training. There could be instances where brain organization goes too far. Focal hand dystonia or musician's cramp happens when the brain reorganizes and the fingers of the musicians who practice extensively become clumsy, fatigue easily, and make involuntary movements. So what happens is that there's extensive reorganization of the thalamus and cortex, so that touch responses to one finger are going to overlap with another finger. So it's going to affect the ability to have control over each individual finger, fine motor movements for the hands. And the result is clumsiness, fatigue, and involuntary movement. So there's a relationship between brain development and behavioral development. Adolescents tend to be more impulsive than adults. Impulsivity can be a problem when it leads to risky behavior. Adolescents tend to discount the future. Adolescents are not equally impulsive in all situations. It can be influenced by peers, the amount of time to make decisions, etc. These things can affect the decisions that are made. If we look at studies that have been done a while ago and we look at heteronormative behavior, we can see evidence that heterosexual males will behave riskier when there's even just a single female present than they will if they're in a group with just other males. The prefrontal cortex of adolescents is relatively inactive in certain situations, but this may or may not be the cause of impulsivity. In old age, some neurons lose their synapses and the remaining synapses change more slowly than before in response to experiences. Brain structures begin to lose volume, but research underestimates older people. People vary in respect to intellectual decline. Older people have a greater base of knowledge and experience and may find ways to compensate for losses. And we'll talk about this more when we talk about plasticity in our next module. We're going to talk about the ability to use other skills, other areas of the brain to compensate for losses. We also see that individuals who exercise more show less decline than those who do not. Okay, so let's end there. Okay, and I'll see you next time with plasticity.